be, they have to limit it to a certain list of 8 practicals. I'll show you that list in a second. For question 9, it's 7 practicals. Question 10, it's another 7 practicals. This is the first of the mandatory practical questions in section B. It has to be this set. Look at this. No, look at these ones. Food tests. That'd be great. You know, you'd be mad not going in there, or you'd be mad to go in there not knowing your color changes like burned into your memory. You really need to. Right. So know all those food tests and what the starting color is, what the final color is, not just the final color changes to whatever color, from this color to that color, from blue to brick red, from yellow brown to blue black. Right. We'll look at those in a second. See the green ones there? They're all ecology ones. They sometimes ask the ecology ones in section B, but here's the thing: they often, because there's a big ecology question in section C, they often include a bit about the ecology survey in there, in section C, in the long questions. But you know, they might not. So you can't be sure. They might just leave the ecology field study questions, those 4 things. There's 4 of them, but do they amount to 4 practicals? Well, they kind of do. They might leave some of that till, you know, capture, recapture, or the quadrat stuff. They might leave it to the section C question. The 2 in purple there, they're not 2 experiments. They're not 2 activities at all. It's one thing. It's examining onion cells and cheek cells under the light microscope. That's the practical. You probably did it all in one double. And you might have spelled it across 2 1-hour classes or something, right? So not hard, but you just need to make sure that you have it down for exam purposes. Like, what are they going to ask, and what can they ask, and what's the answer? What's the answer they're looking for? Then the last one is really interesting. See that isolating DNA from plant tissue? Like, that's the obvious one on the list to go, okay, that's pretty hard. The rest of them are grand. You might not agree with me, but that's what most people think. That's really weird that that's there. I'll tell you why it's weird, because it's totally out of sequence. All of those other ones, and all of the others in the other list I'm going to show you in a second, they're all in order as they appear in the syllabus, right? Except that DNA one. They took that from way down the list and they bumped it up into this question on this list for question 8. Why? Because they reckon it was too easy otherwise. That's what I hear. Because they reckon that there's not much real kind of juicy questions they can ask in there, especially if they don't ask an ecology question, because the ecology question is probably going to be asked in section C, right? So the DNA experiment they ask, because it's got plenty of, you know, it's a juicy question. They can ask lots of different questions to really test your knowledge of cell structure and, you know, your understanding of why you used each of these different things for each of the steps, right? So hasn't been up for a while, that one, isolating DNA from plant tissue, just saying. This is the second list. Demonstrating osmosis, that's Bisking tubing, unless you did potato discs, I don't know, maybe you did. So Bisking tubing, we'll go through in a second. Look at those 4 blue ones. Why are they all blue? They're all blue because they're all enzyme experiments. You'd be mad not to go into the exam knowing your enzyme experiments inside out. Look at the odds. The odds are that it's going to be an enzyme question. Probably not one of those 4, probably a mix of those 4, but like seriously, odds on that it's going to be an enzyme question. But they might not just stick to enzymes. They might ask bits of enzyme questions and bits of photosynthesis question and a bit about the fermentation by yeast to produce alcohol. They might ask a mix of all those. They probably will. But you have to know your enzymes going in there. Like, you really do. This is where the most likely place in the exam is enzymes going to appear. And I guarantee you that enzymes are going to appear in the exam in a big way somewhere. It's either going to be here or it's going to be in section C in one of those Unit 2 questions, the 2 Unit 2 questions. But wherever it appears, you need to know enzymes. This is the last set. And like I said, we're going to fly through these, but you may or may not have done some. I know some teachers are just going, I'm not doing some of those because they've got plenty of choice. So, you know, we've got bigger fish to fry, and I can't argue with that. So, but I won't ignore them because some of them are grand, right? So, but for example, the dissection of the heart, investigating pulse rate, they came up in one of the papers last year, either the deferred paper or just a year before that. They don't come up that often, those ones. So I'm not going to spend any time on those at all. But I'll look at the other ones in double quick fashion. So you see the way it goes. In question 8, there are 8 practicals that can be asked. There's a food test, there's 4 ecologies, 2 are one of them really, right? An examining cells and the DNA extraction. You're going to have probably a mix of those. It could be just on DNA extraction. More than likely, it's going to be a mix. Could be on ecology, and again, it'll be a mix of ecology questions. Let's look at this. These are the 4 tests: starch, fat, reducing sugar, protein. Reducing sugar. Sucrose, this is the starch test, right? Look at the colors, get the colors straight. Yellow-brown to blue-black. You add a few drops. If the food is solid, you can put the stuff, iodine solution, straight on the food, or you can take the food, mixed up in water, add it. What's your control? Text box, please. Answers in the text box. What's your control, everybody? What's the control? What are we going to use as the control in this test? What are you going to say? If you're asked, what was the control when you did the starch test? What are you going to say? Answers, please, folks, in the chat box. I see the answers coming through. What's your control? Careful. I will give you a hint. Almost all of the practicals you do, it's the same control. What is the control in the starch test? Good work. 2 out of 3 right answers so far. Anyone else? What's the control? Last few seconds, last chance to give your answers. Water. So here's the thing about controls. What a control experiment is an experiment that you do in parallel to the test experiment, right? The test is where you get a food, put it in a tube, add water, and add your iodine, right? That's your test. The control is where you remove the thing that you're testing for. In this case, you remove the food. What are you left with? Water. You need to have something that you don't have. That, in other words, you need to remove the thing that you're testing, which is the food, from the solution. What are you left with? Water. That's your negative control. All the controls, and I know I sympathize. So somebody put down like, starch could be your control. Not in Leavens biology. In Leavens biology, controls are always negative, to show that the thing that you're testing, that the reason that you get your result is because of the thing that you're testing. The reason it goes blue-black is because there's starch in the food. Now you might think, well, it'd be a good idea to just check that, that does it work for starch, right? Check your iodine is working and all that. That's fine. That's not your control. Not in Leavens biology. Leavens biology, it's always a negative control. So you need to just remove the thing that you're testing from the experiment. That's your control. Okay? So it's water. It says it right there. It'll stay. It'll stay. What's the control in this? It's water. There's only one spot on this, but actually you should have brown paper. You put your food sample or smear it, butter or bit of meat or whatever, cheese, whatever it is you're testing for fat. You smear a spot of it, or you drop it if it's liquid, onto your brown paper, and beside it you drop a drop of water. That's your control spot. Here's the thing. Both spots will be translucent. What does translucent mean? It doesn't mean that it becomes see-through. You can't turn a piece of brown paper into a window by smearing butter all over it, right? It does become translucent, though. Translucent means that you can see light through it if you hold it up to the light. Here's the thing, that translucent spot, you'd get a translucent spot if you put water onto brown paper too. Try it. But the difference is you need to allow it to dry. That's key. You need to allow it to dry, and then the control spot, the water spot, isn't translucent anymore. But the greasy spot will be. Reducing sugar. Failings or Benedict solution. Probably most of you use Benedict solution. Benedict solution is blue, straightforward blue. It needs to be heated in a boiled water bath. That's kind of misleading there. You don't actually keep boiling it with, you don't need to anyway, keep boiling it with a Bunsen burner. It just needs to be boiling water or boiled water. So boiled water bath, put the test tube in for 10 minutes, not just like heated for a minute or whatever, 10 minutes. It will eventually go brick red. That's the color they want. They don't want any other color. They don't want red, or they don't want purple, or they don't want orange or any other color. They want brick red. Just give them brick red and they'll be happy. Goes blue to brick red. Only for reducing sugar. What kind of things are we talking about? Milk has lactose in it. Orange juice has fructose in it. What else? anything with glucose in it, but not sucrose. Sucrose is not a reducing sugar. Okay. Protein. You need to know that it's a mixture of sodium hydroxide and copper sulfate. So you add sodium hydroxide to make it alkaline, and then you add in copper sulfate, and it'll go purple if there's protein present. Purple, blue to purple. So copper sulfate and sodium hydroxide, that's the Biuret reagent. Okay. So, but you need to know the ingredients on the Biuret reagent, blue to purple. Ecology. This is a simple key, right? No worries. Thank you. If you get dropped, my sympathies, and I hope you're able to get back on again. But thank you for letting me know. This is a binary key. We're only going to be looking at a binary key. So there's only going to be two answers. That's why it's binary, right? So yes, no, or has wings, doesn't have wings. 6 legs or 8 legs. You know, there's only two answers in a binary key. They might give you a binary key. That'd be great. You know, here's a bug, picture of a bug. Here's a key. See if you can identify the bug. That'd be that's like a junior cycle question, though. You know, if you got that, you'd be laughing. So understand what a simple key is, but remember that it's a binary key and be able to explain what it is. You know, it's just a system for identifying animals. If they ask you what a binary key is, for identifying animals where you get two choices, and your two choices eventually lead you to being able to identify the animal. And what's it based on? Based on their physical characteristics. That's what it's based on. It's not based on their behavior. Not the keys we use. They're based on physical, or you know, their DNA or something, right? It's just based on their physical characteristics. That's a binary key. We use it for identifying plants or animals. Okay. If you're collecting animals to identify them, you use your key. Then this is all the ways. They asked the Tullgren funnel last year. People were freaked out. Loads of people had never seen a Tullgren funnel in their life. See the thing on the top right there? Tullgren funnel is basically a big funnel, and you put a gauze on the top, you put a bunch of soil there, and you put a light on top. And the heat and the light causes the small organisms in there to move away from it, and they go down through the gauze into the funnel and down into the jam jar that you've got underneath. So that's a Tullgren funnel. It's for small soil organisms. But generally, what you would have used is a pooter. I hope you got out there to use a pooter at some stage in your life. And you know how the pooter works, I guess. Suck on one end, and you put the second tube on the pooter on top of beside the bug that you're trying to. Not good for slugs or snails or worms or anything like that. Good for small creepy crawlies, small invertebrates. Okay. Beating tray. You can either have a small beating tray that you hold yourself, you know, something that you hold under the tree, and a stick, and you sneak up on the tree and you whack the tree with one sharp whack, and all the bugs come falling down. And you don't just go tapping it, because as soon as you start tapping it, they all cling on, right? So you just one sharp whack, and the bugs fall on your. Well, of course, immediately they're all scurrying away. So ideally, what you want is your pooter there ready to capture them before they all fly away or scutter away. Right? Beating tray. Sweep net, only good for long grass that doesn't have thorns in it, or, you know, not good for like a holly tree or something, right? You're just going to wreck your sweep net. Pitfall trap, though, is important. Pitfall trap has to be a container, not glass, ideally, not sharp, not dangerous, not anything like that. So plastic is good. Tin can is okay, as long as it's not sharp. Has to be so that the mouth of the thing is at ground level. That's important, right? It's not important not having sticking up out of the ground. You can't just dig a little bit of a hole and then stick a tin in it, because things are going to fall into it then. That's the idea, is that as they're walking along, God loves them, especially if you put a piece of wood over it, that serves two purposes. One, stops it filling with rain and they drown, which is not good. But also, if you put a plank of wood over it or something, of course, what do most invertebrates or small, you know, organisms, they all go under that's what they do, right? They go where it's dark and where it's moist for shelter. So they go under there and then they fall in your pitfall trap. Two quantitative habitat plant surveys that you do. They're slightly different. One is easier. This one is the easiest one. This is the percentage frequency of a plant species. You know, the quadrat is just an open grid. No, it doesn't need to have grids in it, or you know, it's not gridded. It's an open square. And they talk about throwing quadrats. You don't throw the quadrat, right? The reason you don't throw the quadrat is because you'll damage plants and whatever, and you'll wipe out a few wood lice on the way. And so you don't want to do that. What you do is you take a pencil, and you throw the pencil over your back. Why over your back? To avoid bias. Because if you're out there surveying and you have a thing for, primroses or whatever, then, and you're counting primroses, well, you're going to go, oh, there's a nice bunch of primroses, and you're going to fire the pencil where there's a bunch of primroses, even subconsciously. So to avoid that bias, you're going to just throw it over your shoulder without looking. And so wherever it lands, you take your quadrat, then you go over to it and you place your quadrat down so that the pencil is in the center of the quadrat. Okay? Then what you do, supposing it's moss you're looking for. So you're on your, grassland or whatever you've surveyed. And if you see a moss plant inside your quadrat, you tick, or X, whichever. It's there. You're only checking to see if it's there or if it's not there, right? And you do that 10 times. And if it's there, if there's moss in 7 of your quadrats, then you know that there's 70% frequency of moss, right? If there's 3, if you found ferns, doesn't matter how many you found, by the way, you're not counting them, right? You're not going to your quadrant and going 1, 2, 3, 4, 6 ferns. That's not what you're doing. You're just going yes ferns or no ferns. That's all, right? And then you count up how many quadrats had ferns in. That's your percentage frequency. Percentage cover is a different thing. Percentage cover is where your quadrat now has grids in it, right? So it's like maybe 100 grids, 100 squares in it. And you do the same thing with the pencil and that. You put your quadrat down. Now you take your pencil and you decide, okay, I'm going to go for the bottom right-hand corner of each square. And you put your pencil in the first square. If it touches moss, right, then you go, okay, that's 1 moss. Next square, no, it doesn't touch moss. Next square, no moss. Third square, yes, moss, that's 2. 3, 4, 5, 6. On you go down through every square till you get to whatever, 25 out of 100 squares, you hit a moss. So you're not seeing if there's moss in each square, you're just seeing if your pencil touches the moss in each square, right? That's the best way of doing it. Number of hits, number of times you hit a moss, literally hit a moss with your pencil, over the total number of squares. So if you got 25 out of 100, that's 25% cover, moss, okay? Population of an animal. This is the capture-recapture method. So here's how it goes, right? You go out with your pitfall traps, set a load of pitfall traps, maybe 10 or something, scattered around the habitat that you're studying. And maybe you're looking for snails. Let's say you're looking for snails, right? So you want to find out what the population of snails is in your habitat. You put randomly scattered, evenly spread out pitfall traps, as many as you can. Let's say 10. Set your pitfall traps. Off you go. Go to bed, come back in the morning, go to your pitfall traps, and you find how many snails that you have. Count the number of snails that you captured on day 1. That's day 1. But when you capture them, for each of these snails, you want to mark them. Now you need to be careful how you mark them. You want to mark them so that they're not like you don't want to put a big fluorescent red target on them, right? Because they're just going to be, local blackbirds just going to come and take out all the snails on you. You want to mark them in a way that you can identify. The mark will last, but it doesn't disadvantage them or make them easy prey. Okay, so something that isn't toxic is going to harm them or make them easy prey, but that will, waterproof, all that stuff. Once you mark them, you release them, not right beside the pitfall trap. You don't want to just go straight back in the pitfall trap, but somewhere, not too far away at the same time. And you want to make sure that they're not, again, that they're mixed evenly around the habitat again, and that they're going to go back to normal as soon as possible. So no stress, no trauma for the snails. Okay. Day 2, you come back and there's a load of snails. You've reset your pitfall traps, gone off to bed again, come back in the morning, check your pitfall traps. Some of the snails in your pitfall trap have the mark on. Some of them don't. Okay. You count the number that you've captured on day 1 already, and you count the number that you captured on day 2, total number, and you count the number that were recaptured. Okay. And this is the formula. You multiply the number captured on day 1 by the total number captured on day 2, times up those together, and divide by the total number of recaptured snails from day 2. Recaptured, the ones with the mark on. So supposing you had 40 woodlice or 40 snails on day 1 and 50 on day 2, but 10 of those 50 were recaptured, 40 by 50, 2,000, you know that there's 200 woodlice, estimate 200 woodlice in your habitat. It's literally way more than that. You know it, woodlice everywhere. So, but that's the idea, okay? So you need to know this formula, you need to know all that stuff, but it's a nice question to do because it's easy to remember. And if you've done it yourself, it's even better. But hopefully that'll give you a good idea if you didn't. This is like a small thing, right? But don't underestimate it. The syllabus wants you to know how you measured in the habitat study 3 abiotic factors. What are abiotic factors, remember? My answers, please, in the chat box. What's an abiotic factor? Abiotic factors, what are they? Good. Oh, we all very good. We know our stuff here. Abiotic factors. So be careful, right? This is where marking schemes can be dangerous. So marking schemes, and you've probably been looking at marking schemes. Marking schemes will give you, and I know you're typing in, chat boxes, so you're trying to be brief. Marking schemes are just keywords. They're not answers. They're guidelines for the examiner as to what constitutes a right answer, but they're not answers. If you were asked what's an abiotic factor, if you wrote down non-living, they're not going to give you the marks. They might, but they're probably not going to. That might be what's in front of them. But what you need to say is it's a non-living factor affecting the life of an organism in a habitat. That's what it is. A non-living factor in a habitat that affects the life of an organism. That is an abiotic factor. You need to give them the explanation. You can't just give them a keyword. Okay. So it's something we got to watch out for, that marking schemes tend to mislead us into thinking, oh, well, that's the answer right there. They're not the answers. They're guidelines to the examiner as to what constitutes right answers. Okay, abiotic factors. I picked these, you can pick any ones you like, right? I picked these 3 because for each one of them, you use a meter to measure the factor, right? So soil pH, you use a pH meter, light intensity, you use a light meter, and you use a thermometer for temperature, right? You can use universal indicator paper for soil pH, but you can remember that it's a meter for each of these 3 things. You do need to understand, though, like give some sort of something about how these 3 abiotic factors affects the lives of plants and animals, examples of plants and animals that you've studied. So you need to be able to give them named examples and how these abiotic factors influence their lives. Okay, so some plants will prefer acidic soil. Good to have an example under your belt, like hazel or oak. Oak prefers acid soil. Willow and nettles prefer alkaline soil. So if you can remember that, that'd be great. Light intensity broadly, you can remember. So think about the things that are like shade. What kind of things like shade? So mosses and ferns like shade. Oak trees like full sunshine. That's why they grow tall and why they throw the leaves up and out into the air, right? So that kind of idea of light intensity, how it affects. And then temperature affects germination broadly. You know that of seeds. So it will only occur if it's suitable temperature. If the temperature is too low, then that will reduce the metabolic rate of most animals, and they won't be able to, move quickly, or they won't be able to hunt effectively. Then they won't be able to, whatever, all those things, and their prey won't be able to grow properly. Plants don't grow properly at the low temperatures, so that's going to affect any herbivores, all that stuff. The other thing is that freezing can break dormancy of seeds. So, that's a useful thing to remember, is that dormancy of seeds is broken by freeze thaw. So now, after a freezing event, you might have germination, and some seeds need that freezing in order to break dormancy. So those kind of things are good to know. Light microscope, not going to go through all the different parts. Well, actually, you know what? Tell me about the eyepiece. What's the eyepiece for? Answers in the chat box, please, folks. What is the eyepiece for? Careful. The eyepiece for. Okay. They may or they may not give you marks for saying it's what you look through, because that's obvious to everybody, right? There's more going on with the eyepiece than just that it's for looking through. The eyepiece has a lens in it for magnification. That's really what they're looking for. So be real careful. So the eyepiece at your standard, higher level, they might expect that it's not just broken through. So you need to know that the eyepiece has a magnification lens, usually a 10x lens, 10 times lens, and in combination with the objective lens, you multiply those two numbers, supposing the objective lens has 40 magnification and the eyepiece has 10. Together, that's a 400 magnification overall for looking through that microscope, through that objective lens, okay? What do the lenses do? They magnify the object. Don't go zooming. They don't do zooming. They won't give you the marks for zooming. They want magnifies, makes it appear larger. That's fine. Magnifies is the answer. What does the coarse focus wheel do? Don't say it focuses, right? Don't say it adjusts, because you're not explaining. What's it adjusting? It sharpens the image. Sharpens the image is the phrase that you want. Thank you, Lillian. The other thing, stage, where you put the sample, the light source, because the light has to go up and through the sample into the lens for you to be able to view it. What's the iris of the diaphragm for? They're kind of slidey bits that you can open and close an aperture. It varies the amount of light that goes up into your sample. That's what they're for. So plant cells, you used a very, you've got the onion, and in between the two layers of an onion, there's a really thin epidermal layer, a dermal layer of tissue. It's, as Lillian mentioned earlier on, it's about 1 cell, maybe 2 cells thick, depending. That's really useful to us because it means that under the microscope, the light will go through the cells and we can see the cells really easily. Much more easily if it's stained with iodine and the nuclei are stained. You need to, when you put it on a slide, that's the thing, you put it on the microscope slide, right? You put a drop of water or stain on it, iodine, and you lower the cover slip at an angle with a pencil, like a drawbridge. Why do you do that? To squeeze out air, because air bubbles are your enemy when you're looking down a microscope. So squeeze out air, and also it keeps the image, it keeps the whole sample flat, right? Why do you add liquid at all? Well, the samples will dry out. If they dehydrate, then their structure gets all distorted, and you're not looking at something, as it should look. So if it dries out, that's not what you want. So the cover slip kind of helps the drying out, but also it keeps it flat. Because if you don't put a cover slip on, and the light comes up through the sample, if it's just a big blob of water like that, then the light will just scatter it left, right, and center, and you'll see, won't see much. Won't be all fuzzy and messed up. So you need to keep the cover slip, but mostly the cover slip is to it's two things: to keep it from drying out, and also to allow the light to move straight up through the sample into the lens. That's what brick wall. Remember, it looks like a brick wall. Each cell, what do you get to see? Not much. You get to see the nucleus, you get to see the cell wall. That's it. Can you see the cytoplasm? Well, it's there. Do you actually see it? Not really. You don't see mitochondria. You definitely don't see chloroplasts. You don't see vacuoles. No vacuoles, right? You're thinking, well, plant cells are vacuoles. Yep, they don't. So you just see a nucleus, and it's stained yellow-brown. Animal cells, what animals do we use? Cheek cells. Swab or finger. 1 minute. Smear it on your microscope slide. Allow to dry. Really important. Totally different to the onion cell where you don't allow to dry because that'll mess up the sample. In this case, you need to allow the gunge that you've just spread. I know it's great, isn't it? Smear cheek mess onto your slide. You had to let that, like a euro-sized coin blob of it. You need to let that dry because otherwise the cells won't stick to the glass. And when you add your water, they just all get washed away. So you kind of need to let that dry, then add your water or your methylene blue, and then lower your cover slip. Same story. View under the microscope. What do you see? These are your cells, cheek cells, nucleus, no cell wall. Do you see the cell membrane? Yeah, not really. You see where it is, edge of the cell. You can't actually see the cell membrane, not really. Look at the right-hand picture. That's a better kind of image. You can just, all you see is like, well, I know where the edge of the cell is because the cells stain kind of, a bit. But really, all you're seeing is the nucleus. Okay, last one. I know I'm keeping you, but this is really important, and I want to finish on this one because this is the first question then in the bag, first of the section B question. So it's really important. Give me 5 minutes maximum, and then next week I'll start with the other hoot. Is that a deal? Okay. Step 1. Whatever plant you used, we used strawberry. You might have had to use onion. If you were really unlucky, you had to use liver. No, it's plant tissue. You didn't have to use liver. If you were unlucky, you had to use onion. It's because the smell, right? It smells of onion afterwards. Chop it up. Chop it up really finely. Why do you have to break down the cell walls? So you chop it up as fine as you can. You then add it to a mixture of salt, helps the DNA clump, and detergent that helps disrupt the plasma membrane. Remember we said that? Not just the cell membrane, but all the other membranes like the nuclear membrane, especially the nuclear membrane, right? That's because the DNA is all contained in the nucleus. So you want to disrupt that membrane as well. But it's the same kind of membrane. It's a phospholipid bilayer. Detergent is your only man for that job. It's better, you know this from washing up, and I know you do loads of washing up. It's better at hot water than cold water, but not, it has to be pretty hot. The reason it has to be 60 and not 40 or 30, it has to be 60 because if it's only warmish, right, what happens is all these enzymes that chop up DNA get released by the cell. It's like a defense mechanism because it thinks it's being invaded by a virus or something. So cells have a defense mechanism where they produce these enzymes that chop up DNA. Yeah, well, they're your enemy in this experiment. You don't want enzymes that chop up DNA because then you'll have no DNA left. It'll all be just chopped up into tiny pieces and you won't be able to find it. So what you want is you want to have the temperature high enough that those DNases can't work. Those enzymes that chop up DNA don't work at 60 degrees. It's too high, right? They're not going to be denatured. You can't boil it. And the reason you can't boil it is because that'll knock your DNA, actually. So what you want is you want just hot enough to keep those enzymes from destroying your DNA, but also warm enough that the detergent can do its job and the DNA is not going to be messed up by it being too hot. So that's 60 degrees. That's why it's 60. You get the idea? So that's 10 minutes, 15 minutes. Those two things happen. DNA gets released because the cell membranes are destroyed and the DNA clumps because of the salt. Now you have to cool that straight away. Same idea here. Cool it on ice quickly as possible because you don't want it to stay kind of warm. If it cools down to like 37, those DNA chopping enzymes will move in and they'll just wipe out all your DNA. So instead, you want to cool it on ice pretty quick. And then while it's still cold, you want to fairly quickly pour it into your blender and you want to do 3 1-second bursts on your blender. Why only 3? Because if you do longer than 3 seconds, and I'd recommend 3 1-second bursts, if you do more than that, the DNA, the long strands of DNA will get chopped into small pieces. Again, that's not what you want, right? You want to keep the DNA relatively intact, some of it anyway. So 3 1-second, but it's long enough. Why are we doing this in the first place? To kind of make sure that we finally, break up those cell membranes and release the cell contents as much as possible. Okay, finally, kind of, you're just trying to release the DNA from the cells. That's why you blend it. Then you filter it, not through normal lab filter paper. That'd be way too slow. You'd be there till the end of your days waiting for the filtrate to go through. It's big, gungy kind of mess in your beaker, So you want actually what you want is coffee filter paper. You know the stuff they put in coffee filter. If you know coffee, most people don't use coffee filters anymore, but you know the things, right? They're grand because the pore size is big. So, but it's not big enough, not too big. So the DNA is now in solution, by the way. The DNA is not, you can't see it. It's in solution, it's dissolved. That will go through in the filtrate and all the bits of cells, cell membrane and bits of nuclear membrane and all the ribosomes and all, they'll all stay in the filter paper. So now you've got filtrate coming through that contains your DNA. It still will have protein in, but hopefully some of the protein will be gone. At this stage, you can add protease to finally wipe out, properly digest all the protein. Because remember, your DNA is wrapped up in protein. Chromosomes are DNA and protein. So the DNA is wrapped up with these proteins that keep the chromosomes together. So you want to get rid of that. Protease it works well. That's protein digesting enzyme. Now you get your filtrate. You need to keep it cold during this whole time, right? Because you don't want those DNases attacking your stuff. Protease will get rid of those DNases, of course, as well, right? Then you have ice cold ethanol. You take your ethanol from the freezer, and the reason it needs to be ice cold is because 2 reasons. One, DNA is insoluble in ice cold ethanol. When it's really freezing cold, the DNA can't stay dissolved in it. So if you take your tube and you gently pour over ice cold ethanol onto the top layer, then what happens is the DNA in the filtrate comes out of solution because it can't dissolve. It's it's insoluble in ice cold ethanol. So at the interface in between the ice cold ethanol, it'll sit on top. The ice cold ethanol is less dense, so it floats on the filtrate, right? And so where the two solutions, where the two liquids meet, you'll see the DNA coming out of solution. Takes a minute or two, happens pretty much straight away, but it'll continue to come out of solution. Then you put in your wooden splint and you fish it out. There's another thing, and be careful about this. I don't know how well this is not. It's a bit misleading in some marking schemes or some textbooks. They say that the reason that the DNA comes out of solution is because the ethanol dehydrates the DNA. Yeah, kind of a bit of a fudge. I would go with DNA is insoluble in ice cold ethanol. There is a, water molecules are removed by the ethanol, by the ice cold ethanol, and so the DNA then floats because it becomes less dense. There's a whole thing going on there, but I just go, it's insoluble in ethanol. That's your best bet. There's notes here. This is on your course. I'm pretty sure it's on your course. If not, then I'll make sure. This is an updated version actually, so I'm going to swap this out. I haven't done it for you guys. I will make sure I do that. So all the different step-by-step reasons, and I think it's really crucial that you should know that. That's what it looks like in the end. So yeah, so looking at these ones, right? We'll fly through these. So that's the last bunch. We'll get to them. So this is what we were talking. Oh, that's the ones we did last week. Flying through. And we're here. So 4 enzyme practicals. This is the Viscon tubing thing. Just yeah, the one on the right is the control. The one on the right. Remember what happens with the Viscon tubing. You take it. It looks like sellotape to begin with on the left hand side. You see, then you wet it, and it turns into a tube. You can open it up and whatever, and you fill it with sucrose solution. You need to do. There's 3 things that are really important with this experiment. One, you need to put the same amount into each one. Why? To make it a fair test. You put the same amount of sucrose into each one. No, you don't. You put, so let me just show you. You put sucrose into one, and you put the equal volume of water into the other one. So the control contains just water. And so what you do is you need to measure the volume of what you put into them, water into one and sucrose solution into the other, or glucose solution. Sucrose is what we usually use. You need to tie a double knot at both ends to make sure they don't leak. And then you pop the 2 kind of sausages into 2 separate beakers of supposedly distilled water, right? But before you do that, you need to measure the mass of your sucrose Viscon tubing sausages, right? You also want to tie them so they're not too turgid, right? So they're not too swollen, because they're going to swell up. At least that one is. The sucrose one is going to swell up. When we go back and we look at the results, the control doesn't change at all. Doesn't change in volume, doesn't change in mass. And the way you, after the results, you put them in and you leave them there for half an hour. When you take them out, you need to dry them off, because you don't want, and you get to pop them on a mass balance, get the mass of each of them, the test one and the control one, and you compare the masses to what the masses were when they started. The control one shouldn't change at all. The other one should have increased in mass. It will also have increased in volume. You cut it open, you pour out the stuff, and you measure the volume of the liquid inside it. But you can also see, because you're observing, one of your results is that it's gone turgid, it's swollen up. And the conclusion is, from all those 3 pieces of information, all that evidence you gather, is that water has moved in by osmosis from the distilled water solution into the sucrose because of the concentration difference. Osmosis, remember, 2 things you need to remember. It's the diffusion or the movement of water molecules from an area of high water concentration to an area of low water concentration. And sucrose solution has a relatively low water concentration compared to, the pure water outside in the beaker. But the second part of osmosis you have to put in your definition is it's through a selectively permeable membrane. That's osmosis. If it's not going through a selectively permeable membrane, it's not osmosis. If it's not the movement of water, it's not osmosis. Only water moves by osmosis, and only water moves by osmosis through a selectively permeable membrane. This is the enzyme experiment. So you're keeping the temperature constant because you're changing the pH. The variable you're changing is the pH. Everything else must stay the same. The volume of the celery juice you put in there containing the catalase, the volume of the hydrogen peroxide substrate that you're putting in there, the temperature must be kept constant. That's your water bath coming in. And the only thing that you're changing is the pH. So actually, you're going to have 4 setups like in your picture here. One with a buffer, and the volume of the buffer must be the same in each case as well, right? pH 4, pH 7, pH 9, pH 12, usually 4. And after you mix all the things together, bar 1, you either have to leave out the enzyme or you have to leave out the substrate. So either leave out the celery juice or the hydrogen peroxide until you're ready with your clock. Then you add the last thing, and then the reaction starts. And the foam will rise, and you have to count, or you have to measure the time. No, you don't. You measure the volume of oxygen foam, right? Trapped oxygen that's produced in 1 minute. Or if it's really slow, maybe in 5 minutes. But it should be, in 1 minute or even in 10 seconds, depending on how quickly your reaction's happening. It all depends on how concentrated your hydrogen peroxide solution is. Okay? The temperature that you would use would be fine, 20 degrees. That's usually good for something like celery. 20 degrees is fine. If you were doing, so this is your, they're not measuring cylinders, but you get the idea. It's the volume of foam. You can measure height of foam as height of oxygen in foam containing oxygen, right? Don't forget the oxygen bit. And then the similar thing when you're looking at the effective temperature. So you would need, going back to this here, you can have the same water bath with 4 cylinders in. I mean, in reality, who really uses a water bath, Everyone does this just on the bench. I would imagine. Did anyone really use like multiple water baths or a big, huge water bath? No, we just did it at room temperature. But that's not going to get you through the exam. If you're asked in an exam, how did you keep the temperature constant? You said, we used a water bath. You lie. If you're changing the temperature, then you have to use a water bath. Now, your water bath might have just been a big beaker that you just took some water from a kettle and you put it in, pour, cooled it down with a bit of tap water, measure the temperature, went to 40, there we go, that'll do the job. Put your cylinder in, let it warm up to the temperature. Check that, with the thermometer that your mixture in your cylinder is now the temperature that you want it to be at. And then you add your last ingredient, which is your celery juice or your hydrogen peroxide, right? And then again, start the clock, measure the volume of oxygen containing foam after 1 minute. That's your rate of enzyme activity. You do that at different temperatures and you get that kind of graph. What's the optimum temperature? 20 degrees for catalase activity. Okay. The last thing you do then is a similar experiment, but you have to be careful with this. This is easy to kind of mix up. This is you're just doing 2. And what you're checking here, the test is what's the effect of boiling it, right? So you have one that's unboiled. So leave the one that's not boiled for a second and you boil one. Now it's really important that when you boil it for 10 minutes, you allow it to cool down, right? It has to be cooled down because it won't work. Nothing will work at 100 degrees centigrade, your experiment. So you have to cool it down to see if there's any activity left. Because remember, the enzyme only works at 20 degrees, so it won't work anyway at 100 degrees. What you want to see is it destroyed at 100 degrees? It only works it works best at 20. So you cool it down to 20, then you check to see if it works. Of course it doesn't work. Why doesn't it work? Because it's denatured. But the one that you didn't boil, that'll work fine. Keep the pH the same, use pH 7 or 9. When you're doing the pH experiment, by the way, you do find that the optimum pH is about 8 or 9 for catalase. They'll accept like 7, 8, 9. Okay. Investigate these. Okay, that's the heating nitrogen. The last of the enzyme ones. So this is a nice one. So you would have taken yeast cells, a packet of dried yeast, right? And you would have mixed it, or your teacher would have mixed it with sodium alginate solution, kind of like a syrupy sort of gloopy stuff, right? And you would have mixed the two, the yeast slurry and the sodium alginate slurry, gel, liquidy kind of syrupy stuff. Mix them together to get them to mix nicely, evenly mixed, right? Then you draw the gloop up into so gloop is not an exam term, right? Sodium alginate and yeast mixture up into syringe, whatever, 10 mils of it. And then from a height, my glass, and this is calcium chloride solution in here, right? From a height, about 15 centimeters, you need to get the drops to drop reasonable distance so that they form spheres as they hit the calcium chloride. The sodium alginate reacts with the calcium chloride and the beads harden. And what that does is it traps the cells inside. So the yeast cells are stuck inside this gel. And we're not interested in anything that the yeast cells do except that they produce sucrase or invertase, the enzyme that breaks down sucrose into glucose and fructose. So you leave the beads harden for 10 minutes, right? Then you rinse off, you pour them into a sieve and you rinse them under the tap. Two reasons you rinse them. One is to remove unbound enzyme. That's important. Any free enzyme needs to be washed off the beads because it's not immobilized then, right? So rinse off any free enzyme. Also, sodium chloride is formed when calcium chloride reacts with sodium alginate. You get sodium chloride formed and calcium alginate, actually. So sodium chloride is salt. You want to get rid of that as well. But mainly it's about getting rid of the free enzyme. Once that's been, rinsed, then you put them into a column, some sort of a tube or whatever. You might have put them into a funnel, I don't know, some sort of a yoke. And then you warm the beads and you warm your substrate, that's sucrose solution, and you pour it in the top. And you allow it to trickle down through the beads. And as it goes past the yeast cells, they'll take the sucrose and they'll break it into glucose and fructose. And out the bottom comes a mixture. Well, actually, I mean, at the bottom still comes sucrose. The beads aren't that good. Like the yeast are not able to literally convert all the sucrose into glucose and fructose. But enough glucose and fructose comes out that you're there with your glucose test strips. Remember them? Dipsticks that you just test to see what comes out the bottom, and lo and behold, you get glucose. Effective light. Here's the ruler thing, right? The ruler, watch out, the ruler is a bit of a red herring. So the only reason you might use a ruler is because then you can just figure out what the light intensity is. You can write it down for 10 centimeters, 20 centimeters, 30 centimeters, 40 centimeters, and then you can just move your beaker back and forth to those distances, right? But you're not measuring, you don't record the distance for your results. What you plot is you're plotting, sorry, this, oh, try that again. I'm going to have to take away that widget thing that I've got. So on the bottom, you have light intensity on the x-axis. And on the y-axis, you've got rate of photosynthesis. That is literally bubbles of oxygen per minute. Okay? So you're counting the bubbles per minute. But the per minute thing is really important. And you're putting that against light intensity. So you're not putting it against distance. Okay? That's the thing to remember. So I think we've talked about everything else. Oh, a couple of things. One, you want to keep this water bath at 20 degrees. That's what Eladia will like. Eladia. And the other thing you want to do is you want to add as much carbon dioxide to the water as possible. Now, how do you do that? Not soda stream. You add sodium bicarbonate into the water. Or else you actually just use pond water. Because if you use pond water, then there should be enough carbon dioxide in it from if it's a healthy pond, from all the respiration of all the bugs that are in there, like the water beetles and all those kind of things, animals respiring. So pond water is actually pretty good. But if not, then you just add baking soda, which produces bicarbonate ions, which is effectively the same thing as carbon dioxide in the water. Okay. Why do you need that? So that's not going to be a limiting factor, so that they're going to be able to photosynthesize fully as long as they have enough light, because they need carbon dioxide as well. So you make sure they have plenty of carbon dioxide or carbonate ions. This is the alcohol fermentation one. So sorry, bear with me there. This fermentation lock. So the couple of things here. You want to make sure that your glucose solution is sterile to begin with. Your control is going to be glucose only, no yeast. You need to have sterile conical flasks. How do you sterilize them? You can use sterilizing fluid, like Milton's, for like sterilizing babies' bottles and that kind of thing. So you sterilize the flasks, you sterilize the fermentation lock, sterilize the stopper. You have sterile glucose solution. You just autoclave that, right? So it's all sterile. In other words, there's nothing going to grow in there. Then you add yeast, sachet of yeast, and your control has no yeast because you're looking to show the production of alcohol by yeast, right? So you want to make sure that if you do get alcohol, that it's because of the yeast and not just because of some random other thing where, you leave, imagine if that was the truth. You just leave water and it turns into alcohol. if you just leave it there for 24 hours. That would be a different world we'd be living in, wouldn't it? So glucose will get converted into alcohol by the yeast. Temperature has to be kept at 30 degrees. That's the optimum for yeast. And the fermentation lock is, anyone know what that's for? Answers in the chat box. What is the fermentation lock for? Two things, really. Well, one thing, actually, really. What's it for? What's the purpose of the fermentation lock? Excellent. Okay. It's really twofold. So what it does is, so as the yeast are respiring, right? They produce carbon dioxide. Now, they will do normal aerobic respiration for a while. Then they'll switch to anaerobic respiration as soon as they run out of oxygen. So what happens is the oxygen in the air that's in there already gets pushed out because the carbon dioxide that's big. But the key thing is what you've all said, which is really good. No more oxygen can't get back in because it's a valve system, right? But the other really important thing is it prevents, oh, it actually says right there, prevents entry of air. Okay, I take it back. The other thing that is really important is it prevents not just oxygen getting in, but also contaminating bacteria, right? So in other words, it allows the carbon dioxide to get out, it prevents oxygen getting in, and it prevents bacteria getting in. Bacteria can't move this, can't swim or whatever, right? Excellent. Oh, sorry, the test. We've done the lime water test. The iodoform test is potassium iodide, sodium hypochlorite. You get yellow crystals or yellow precipitate if there's alcohol. If not, stays clear. Next question then. I'm going to fly through just a few of these. The leaf yeast, I think, is a good one to know. I'm not going to do the heart dissection and the pulse rate with you. I'm not going to do the auxin one. That one, oh, sorry, I am going to do the auxin one with you. The seed germination ones are dead easy. This one we looked at already, the Vaseline leaf discs. We talked about how you collect them with a sterile forceps and everything. Ash leaves is the usual thing, but lots of leaves have leaf yeast on them. The leaf yeasts are growing on the underside of the leaf. So when you're attaching the leaves to the lid, you want to make sure that the underside of the leaf is pointing toward the malt extract agar. Put them in the incubator for 24 hours upright, then turn them over for another couple of days. What kind of temperature? 20 degrees. That's what leaf yeast is like. When you come back, what do they look like? Oh, where's the picture? There we go. They're pink, orangey pink colonies. They're really nice. That photograph doesn't do them justice. I have nicer photographs. I'll find nicer photographs. Aseptic technique is crucial. That's what we just talked about, right? Trying as far as possible to exclude microorganisms, especially pathogens. They need you to say the especially pathogens bit for some reason from your petri dishes. So what you do, wear gloves, disinfect your workbench, face mask, use sterile forceps, all that stuff. That's all part of aseptic technique. Okay. This is the die cut stem. We already talked about that. Backed blade, slice, thin as possible. Stick it under there's no stain. You put it under, and you get this. What was it again? Let me see if I can remember. Die cut disc and monocot messy. Love that. So this is a die cut. So they're arranged in disc in a ring-like fashion, right, underneath the epidermal layer. They're called vascular bundles. Remember, in a vascular bundle, what does that vascular mean? It means transport tissue. Two types of transport tissue. You got the phloem on the outside, and on the inside, you've got the xylem. Phloem for carrying sugar, sugar solution, sucrose usually, and then xylem for carrying water and minerals. That's the heart, not doing it. Not doing that. IAA, this is an auxin, right? So what you do is you get a clear, I don't know if you've done this experiment, if you haven't done it, I'll just run you through it quick. So radish or mustard or whatever, small seeds. You get a clear 1 centimeter grid, plastic, and you put that on the lid of a bunch of petri dishes, 5 usually, and then you put 5 seeds, cress, radish, mustard, whatever, right? And you put a disc of filter paper on top of the seeds, and then you add distilled water to one of the dish, and one of the dishes, that's your control. That's no auxin. IAA is no auxin. It's indole acetic acid, right? And then you have a series of dilutions that you were probably given that you don't have to know how they were made. It's just a wide range of very different dilutions of this auxin, right? So there are probably tenfold differences in the dilution. So like one would be 10 times as dilute as another, and another would be 10 times more concentrated as another. So you'd have this range of tenfold dilutions, 4 of them usually, and you put that onto cotton wool on top of the filter paper, and you put the lids of the petri dishes onto the bases, and then you put them sideways. Put the petri dishes sideways. Right? Usually you put petri dishes like that. These ones you put sideways, and you put them sideways, and the whole idea of the cotton wool and all the rest of it is to make sure that the seeds don't just fall down when you put them in. You put your petri dishes like this. You need some sort of a rack to keep them upright, right? The reason we do that is because you want the shoots to go one way, and that's phototropism and geotropism will cause the shoots to go that way and the roots to go this way. And the reason you have the acetate grid is so that you can, because they're 1 centimeter, it helps you measure without a ruler, estimate the lengths of the shoots and the lengths of the roots, right? So you give them a few days at 25 degrees, standing like this, and then you come over and you measure the percentage increase for each of the different concentrations. But the basic idea is that low concentrations of auxin promote root growth. Higher concentrations of auxin promote shoot growth, but not root growth. And very high concentrations of both inhibit both. Okay? So low concentrations give you only root growth. Higher concentrations give you only shoot growth. And very high concentrations, very concentrated auxin gives you inhibition. So auxins can be used as herbicides, right? Because they just stop growth if they're in high enough concentration. That wouldn't be good. And then the last two are the seed ones, right? This first one, you don't have to use an anaerobic jar. Perfectly acceptable to have done all this in test tubes. And the anaerobic jar one, instead of an anaerobic jar, you would just have cooled boiled water, if you've done this, and you put oil on top of the cooled boiled water to make sure oxygen doesn't get in, right? And lo and behold, that seed that's underwater with no oxygen doesn't germinate, funnily enough. And the seeds in the fridge, even if they have oxygen and they have a suitable temperature and oxygen and water, they won't germinate. The ones in the warmth that don't have any water, they won't germinate. The only combination that's going to of these different ones is going to germinate is the control. The control is the one that you haven't taken anything away from. Weirdly, actually, what you've taken away from the control, because remember, it's a negative control. Negative controls are where we take away the thing we're testing. So what we're testing is the effect of water, oxygen, and temperature. So you take away, what you're doing is you're taking away the absence of those, if you like. Think about it that way. So your control, be careful, right? The control is the one that you give everything to, because you're not taking away you're not taking away the absence of water. You're not taking away the absence of oxygen. You're not taking away the absence of temperature. You've done that with each of the test ones. The control is the one where you don't not take those away. Last one, broad beans. Soak them the night before. Why do you need to soak them? Why do you need to soak them? Answers, please. This is your last question for this evening, folks. Why do you need to soak the broad beans before you use them the next day? I know it says to promote germination. Why, though? How does that promote germination? What are you doing to the seeds that allows them to suddenly be able to germinate? Oh, good guesses. Excellent answer, Lillian. I love that. It's to break dormancy. If you haven't done seeds yet, break dormancy. That's what you do. So we've done an experiment. We've done a lesson on this. If you haven't done it yet, or you haven't, it'd be worth just having a quick look at it. I would recommend it. Plant reproduction. It's a good one. And it's going to be there, and you don't want to get caught out with it appearing in the middle of your favorite Section 8 question on something else, and they ask you about dormancy. So dormancy is despite the conditions being suitable, that a seed doesn't germinate. Now, to break dormancy, It's like it's a period of really low metabolic activity and no growth. And the embryo stops growing. And in order to break dormancy, various things need to happen. Soaking them usually works. Cracking them sometimes. Freeze thaw naturally out in the wild. Usually, during the winter they freeze, they thaw, they freeze, they thaw. That kind of allows water to get in to the seed, and then once water gets in, then they will germinate. So you soak them, then you take them and you boil half of them, boil a couple. You don't, that'll kill them, by the way. Then you don't boil another couple. You take a backed razor blade, you split the beans down the middle, broad beans. They're like big baked beans, but bigger. And you split them down the middle. What you get then is those two halves are actually cotyledons, right? These are non-endospermous seeds. They have this food store in the cotyledons, in the seed leaves. But you should see a little embryo there as well. So anyway, you put the two halves of the beans down on your starch agar plates. Either they're boiled and dead or they're not boiled and you put them down. The ones that aren't boiled, the live ones, they will produce, because you've started germinating them by soaking them, they will produce, and they'll continue on the nice moist agar, right? They will produce amylase, enzymes, because now you've broken dormancy, they start to germinate. One of the stages in germination is that the embryo starts to produce digestive enzymes to break down the food store. And you've given them extra food, you've given them starch, so they're going to be delighted, these half raw beans. And the embryo is going to produce amylase, it's going to break down the starch. So what happens is, when you come along with your iodine a couple of days later and you flood the plate with iodine, the plate will go black wherever there's starch, but there won't be any starch around the broad beans because it'll all be digested by the amylase produced by the embryo because it's germinating. So to show that there's digestive activity during germination, you should see a clearing zone where it'll be yellow-brown, and around the not clear zone, a yellow-brown zone around the bean, and everywhere else on the plate that where the bean isn't, and the amylase can't diffuse out that far, you get black because the starch is still there. I know it looks like a fried egg, but actually that's a broad bean, and that is how that's the starch agar is all black around the broad bean, and it's not very yellow brown there, but you can see it's kind of yellow around the broad bean. And folks, that is that.